कैमरा काम नहीं कर रहा? अच्छा मैं दिख रही हूँ? अच्छा अब तो ऑफ हो गया ना? अच्छा ठीक है।
तो कैमरा ऑन करना है नहीं करना?
मैं करती हूँ।
चलो आप कर लेना। आप सर फिर कब तक आएंगे?
आएंगे, मैं फ़ोन करती हूँ सर को।
चलो मैं 4 बजे आऊँगा फिर सीधे।
आप अभी 4 बजे आओगे?
तो कंटिन्यू रहेगा ना आपका, तो 4 बजे तक।
मेरा 1 ही घंटे का है। आप 3 बजे आ जाओ फिर से। कहीं जा रहे हो आप? मैं फ़ोन कर रही हूँ आपको भी।
नहीं, क्योंकि सर तो आएंगे, सर तो अपना आप ही कर लेंगे, तो उसमें कोई प्रॉब्लम नहीं है।
ठीक है, देख। Good afternoon, everyone. Are you there? Okay. Okay, sorry for the delay.
No problem, ma'am. No problems, ma'am. No problems.
Okay. So, have you read yesterday the first chapter of Guyton? Have you started reading for hematology lectures?
Yes, ma'am. We read. We have some doubts.
This conference will now be recorded.
Okay, you have doubts? ठीक है. Then those who have not read, Akram, I don't know who is Akram. So please start reading. Actually, you should read the chapter before coming to the class. Okay, so before the class, please read it so that you will better understand it. So I will clear your doubts at the end. Okay, so first we will start the today's lecture. So today's lecture is principles of hematopoiesis. Now, do you know what is hematopoiesis? Anyone knows what is hematopoiesis?
Formation of blood.
Okay, very good. So it is the formation of blood. So yesterday we have seen what is the function of blood, what is the function of different cells. So today we will see how these cells which are present in the blood, they are formed. So basically, where these cells are formed? Do you have any idea where the hematopoiesis takes place? Okay, in the bone marrow. Any other place can you think of? If the bone marrow is sufficient, it is liver. Okay, so if the hematopoiesis, okay. Okay, very good. So if hematopoiesis takes, okay, we will discuss it in detail. So if the hematopoiesis takes place at other places other than bone marrow, then it is called as extramedullary hematopoiesis. Okay. So now let us see about hematopoiesis. So okay, so we will cover this topic under these headings, that is what is hematopoiesis, what is the hematopoietic organ, what is hematopoietic stem cell, then hematopoietic inductive environment, then theories of commitment, then hematopoietic growth factors, and what is the significance of this hematopoiesis. Okay, now what is hematopoiesis? As you have told, that is, it is the formation of blood cells, and there is formation of large number of blood cells, around 10⁻11 cells will be formed each day. So it is a very active organ, okay, because cell division takes place continuously, so that around 10⁻11 cells will be formed each day. Why so many cells have to be formed? Because the lifespan of these cells is very short. For RBC it is around 120 days, but for WBCs and for the platelets it is in some days. Okay. So to maintain that turnover, the bone marrow has to work very actively and it has to form large number of cells each day. So now how is this hematopoiesis, what is the meaning of hematopoiesis basically? Even though it is the formation of blood cells, there are various processes involved in this hematopoiesis, and the first step is the self renewal of hematopoietic stem cell. So do you know what is stem cell? So there are different types of stem cells: pluripotent stem cell, totipotent stem cells. So here we are dealing with hematopoietic stem cells. So stem cells means these cells can give rise to different types of cells. So a single hematopoietic stem cell can lead to formation of RBC or WBC or platelet or monocyte, lymphocyte, neutrophil. Any cell can be developed or can be formed from this hematopoietic stem cell. So the first step is self renewal of hematopoietic stem cell. Now as we know that we need all these blood cells throughout our life, and daily there is formation of large number of cells. So if there is no self renewal of hematopoietic stem cell, then the stock of the hematopoietic stem cell which we have, it will get exhausted and we will not have any more hematopoietic stem cell to form other cells. So it is very important that the hematopoietic stem cells get renewal of themselves, and then they will be stored, and some cells will be utilized for the formation of different types of cells. So the first step in hematopoiesis is self renewal of hematopoietic stem cells, then proliferation of hematopoietic stem cells, then commitment of hematopoietic stem cell to a particular lineage that we will discuss, and then maturation of this lineage committed progenitor cells into mature blood cells. So we will discuss in detail what are the different processes involved in hematopoiesis. But what is the prerequisite required? What is the requirement of hematopoiesis? Can it take place in the muscle or can it take place in the GIT? So it cannot take place at other places because it needs a particular environment. It needs particular growth factors which can lead to survival of these cells as well as proliferation and differentiation of these cells. So these are the growth factors which are required. So they need a particular environment, and that is called as hematopoietic inductive environment, and this environment is provided by in the adult life by the bone marrow and also by the liver or spleen. So at only these few organs hematopoiesis can take place because they get the hematopoietic inductive environment at these places. So in general, this is the hematopoiesis. So we have hematopoietic stem cell, then it can be, so this is the hematopoietic stem cell which can give rise to any type of cell. Now this arrow shows that first it gets self renewal, also it proliferates so that the number of hematopoietic stem cell increases, then it gets differentiated into myeloid stem cell or lymphoid stem cell. Now this myeloid stem cell, this is also a stem cell. Even though it is committed towards myeloid side, still it is a stem cell and it can give rise to multiple types of cells. Okay, so then this again gets committed to either megakaryoblast, proerythroblast, myeloblast or monoblast, and from this stage they will get committed to a specific type of cell. That is, proerythroblast, they will form only erythrocyte. Megakaryoblast can only form platelets. Okay, so now they cannot form either stem cell or any other type of cell. They are committed to a specific cell type. So they are called as committed cells. Like that, lymphoid stem cell can give rise to lymphoblast, and which can again differentiate into natural killer cell or lymphocytes. So these are the different types of blood cells. So the major portion is formed by the RBCs, granulocytes, platelets, and also by lymphocytes, which are again two types, lymphocyte T and B lymphocytes. Now let us see how you already know that hematopoiesis, even though during adult life it takes place in the bone marrow, but from birth or before birth, what are the places where this hematopoiesis takes place? So 6 to 8 weeks of gestation, so you know what is gestation, and okay, so you will learn it in detail during anatomy lectures or embryology lectures. So first the hematopoiesis starts extraembryonic at the extraembryonic place, that is yolk sac. Okay, so first it starts at around 6 to 8 weeks of gestation, and there are few blood islands where the hematopoiesis starts, and that is called as primitive hematopoiesis. We will see what is primitive hematopoiesis. And during this period, the RBCs which are formed, they are nucleated RBCs. So in the last lecture we have seen that RBCs are the cells which don't have the nucleus, but those which are formed during this 6 to 8 weeks of gestation, that is during primitive hematopoiesis, they have the nucleus. Now after 6 to 8 weeks, you can see in this diagram also, which is on the right side. Now after after this period, initial period, the intraembryonic hematopoiesis will start, that is called as definitive hematopoiesis, and this takes place at the it is called as AGM, that is aortogonad mesonephros, and also at the ventral portion of the dorsal aorta, ventral surface of the dorsal aorta, which is not shown in the figure, but we have a next slide for it. Then the after some time, AGM is aortogonad mesonephros. Okay, then after some time the stem cells move to the placenta, where the expansion of stem cells takes place. They proliferate profusely and the number increases. Also they move to the liver and spleen, and during this mid gestation period, the hematopoiesis takes place in the liver and in the spleen, and the RBCs which are formed in the liver and spleen, they don't have the nucleus. Now towards the end of gestation, most of the hematopoiesis will shift to the bone marrow, even though it is taking place from the mid gestation in the bone marrow. Towards the end, it is the sole organ which is forming the RBCs. So after birth, the hematopoiesis takes place mostly at the bone marrow. So during normal steady state conditions, hematopoietic stem cell reside mainly in the marrow cavity, but under certain stressful conditions, suppose there is hemolysis, that is if there is some defect in the RBC membrane, okay, or hemoglobin, and it leads to damage of the RBCs and more hemolysis, then in some conditions the hematopoietic stem cell can migrate and colonize other organs such as liver and spleen, and they can start developing there. So that is called as extramedullary hematopoiesis. So hematopoietic organs, these are the sites. So the hematopoietic organs are these organs keep on changing from prenatal period to the postnatal period. So site changes during prenatal development also. Initially it is yolk sac, then it moves to the aorta, then liver, spleen, placenta, also bone marrow, and then after that it will permanently shifts to bone marrow. Then hematopoietic tissue arises from mesodermal germ layer in the embryo at the time of gastrulation. So in the late gastrulation, two types of hematopoiesis are established. First is primitive hematopoiesis. So it arises in the blood islands of the extraembryonic yolk sac, where visceral endoderm induces endothelial and blood cell development in the adjacent mesoderm. And definitive hematopoiesis arises later, and it begins in the splanchopleura or mesonephros of the developing embryo. This is intraembryonic, this is extraembryonic. So what is the, so this is the cross section. So it is a schematic diagram basically, and this is the dorsal aorta. So I already told you that even though the hematopoiesis starts in the extraembryonic tissue, that is yolk sac, that is a primitive hematopoiesis. Okay, and it will provide the required blood cells to the developing fetus or developing embryo. Okay, so after few weeks the hematopoiesis starts intraembryonically, and there are evidences that it starts at aorta, gonad, mesonephros and at the ventral surface of the dorsal aorta. So this is the schematic diagram showing ventral surface of the dorsal aorta, and these are the cells which are formed from the hemangioblast. So hemangioblast can give rise to either blood cell or endothelial cells. Okay, so they are called as hemangioblast. So it is proposed that the definitive hematopoiesis starts in the ventral surface of the dorsal aorta, and then this hematopoietic stem cell, they shift to the placenta, liver, spleen, bone marrow, and then at these sites the hematopoiesis will start. So what is the difference between primitive hematopoiesis and definitive hematopoiesis? So primitive hematopoiesis is transient, only it will take place only from day 19 to 8 weeks. So mostly it is required for the formation of RBC, but other cells are also formed like platelets, macrophages and lymphocytes, they can be formed. And the erythropoietin, which is the main factor which is required for hematopoiesis, is not much needed for primitive hematopoiesis. And the hemoglobin has different globin chains. We have discussed in the last lecture that it has heme part and globin part. So alpha and beta families of globin chains are formed during primitive hematopoiesis, so it will form alpha 2 and beta 2. Okay, these are the globin chains are present in the primitive hematopoiesis. EPO is erythropoietin, okay, which is secreted by the kidney and which is very essential for the hematopoiesis. Now what is definitive hematopoiesis? So it starts after 8 weeks. We have seen that it is at aorta, gonad, mesonephros or ventral surface of the dorsal aorta, and it begins after 8 weeks and it will lead to formation of all cells. So it lasts throughout the life, definitive hematopoiesis. So it forms all the types of cells, and the RBCs which are formed during definitive hematopoiesis, these are the non-nucleated RBCs, and it requires erythropoietin. So kidneys secrete erythropoietin. If the kidney is normally also it secrete erythropoietin, and if the kidney is exposed to the hypoxia, then large amount of erythropoietin will be secreted by the kidney. Okay, and that is required for erythropoiesis. Now if there is any disease of the kidney, or if the kidney is removed, okay, or if there is any chronic renal failure, then the patient develop anemia because the erythropoietin is less or it is not developed by the kidney. So hematopoiesis will be affected in such patients. So if you have a patient who has chronic renal failure, so it is very important to monitor the hemoglobin level and RBC count, because often we observe that they have the anemia and it is required that they have to be given erythropoietin injections. So we give erythropoietin injections at specific interval to these patients so that their RBC count and hemoglobin level RBC count will be maintained within normal limits. Now during definitive hematopoiesis we have both fetal and adult forms of globin chains. So we will discuss it in detail during subsequent lectures, what are the fetal and adult forms of globin chains. Okay, so it is already discussed that during definitive hematopoiesis there will be rapid seeding of liver by the hematopoietic stem cells, and that is a major site in mid gestational period to form the RBCs and other types of cells. Along with liver, spleen also gets involved in hematopoiesis, but during the latter period bone marrow takes over all the function of hematopoiesis, and in postnatal period bone marrow is the exclusive site for the hematopoiesis. And first initially the hemoglobin which is formed, that is the fetal type, but after birth it gets converted, it will be switched to the adult type. Okay, we will discuss it in during next 2 to 3 lectures. So rapid seeding of the liver means the hematopoietic stem cells which are present in the aorta, gonad, mesonephros, or in the dorsal surface of the ventral aorta, ventral surface of the dorsal aorta, they will move from this space and they will go to the liver. Okay, because they have the hematopoietic inductive environment which is provided by the liver, by spleen, or by placenta, so that they can multiply there, and that is called as rapid seeding of the liver. Rapid seeding of the placenta, liver and spleen will take place, where there will be proliferation of hematopoietic stem cells, so that their number will increase, because we need hematopoietic stem cells throughout our life. Okay, even though most of the hematopoietic stem cells are present in the dormant state, but we need them throughout our life to maintain the hematopoiesis. Okay, because each day 10 raised to 11 number of cells will be formed, so we need hematopoietic stem cells. So they will first shift to the liver, then spleen, and then to the bone marrow. So even though we say that after birth hematopoiesis takes place mostly in the bone marrow, but the cellularity of this bone marrow will keep on changing, or it will go on decreasing throughout our life. So in this diagram you can see that at birth all the bones have almost 100% cellularity. All the bone vertebra, sternum, ribs, femur, tibia. So at the start they have 100% cellularity, but as the age increases the cellularity drastically goes on decreasing. So at the around age at around 20 years of age, so tibia, most of the cellularity in tibia it reaches to 0%. So as age advances from the around 30 years, so the only the axial skeleton which consists of vertebra, sternum and pelvis, ribs, they only continue to form the RBCs, and the proximal end of femur and tibia, they are involved in hematopoiesis. Otherwise the bone marrow, it will be occupied by the fat tissue, so that the function of bone marrow is not now, it is the not hematopoiesis. Only the few places like vertebra, sternum, ribs, femur and proximal ends of femur and tibia, at these places hematopoiesis will continue throughout the life. Now we have seen that bone marrow is involved in hematopoiesis. Now what are the tissues or structures present in the bone marrow which are supporting this hematopoiesis? So this is the, on right side we have one diagram. This is mostly for the maintenance of hematopoietic stem cell. It is also called as hematopoietic stem cell niche. Okay, so what structures you can see in this diagram? So on left side you have whole bone, and from that bone they have taken small part and then it is expanded. Okay, so on left side there is a bone surface which consists of osteoblast, osteoclast, and on right side this is the marrow cavity. So it has the bone marrow. Now if what is present in, so it is an anatomical 3D organization of the different tissue types and their components. So we can see here that it has arteriole, it has sinusoids, then extracellular matrix, and then there are megakaryocytes, there will be adipocytes will be present, lymphocytes, endothelial cells which are lining these capillaries. So all these structures which are present in the bone marrow, they are involved in this hematopoiesis. So they play different roles. So it is important for maintenance of hematopoietic stem cells, their proliferation as well as differentiation. So here you can see the red color cells, these are the hematopoietic stem cells, and there also we will have the progenitor cells and immature cells as well as the mature cells. If you see during your practical, you will see the slides having bone marrow slides. In that slide you can see the different progenitor cells at their different maturation steps, and also the mature cells which are present in the bone marrow. So during your practical you will see the bone marrow slides. So now we have seen where the hematopoiesis starts, from how it shifts from yolk sac to the bone marrow. Now what happens in the bone marrow? Now we have seen that we start with the we start with hematopoietic stem cell, which can give rise to different types of cells. Now this progeny of hematopoietic stem cell, it begins to differentiate. So then it will form committed progenitor cell. Now if we go to the committed progenitor cell, it cannot go back to the hematopoietic stem cell. So, but these early generations of progeny committed cells can give rise to descendants which represent multiple lineages. So these are the progenitor cells. So this is uncommitted stem cell, which is called as hematopoietic stem cell. This can give rise to any type of either erythrocyte, platelet, neutrophil, any type of cell. Now this uncommitted stem cell give rise to, or it can differentiate into committed progenitor cell. Now this committed progenitor cell also has capacity to give rise to multiple lineages of cells. Like this committed progenitor cell can give rise to erythroblast or megakaryoblast. Okay, so different lineages can be developed from this committed progenitor cell, but this cannot give rise to hematopoietic stem cell. Okay, now and the descendants of the multilineage colony forming cells are ultimately restricted to a single lineage potential, and single lineage potential cells, if we multiply them into the different culture media, they give rise to formation of colonies. Okay, so now they get committed to a single type of cell, and then they divide, they multiply, and then they get mature and they form a specific mature form of cell, either erythrocyte, platelet, neutrophil, monocyte, basophil, eosin. So these all cells then can be developed in the bone marrow. Okay. Okay, so what are the terminal phases of differentiation? So in the final stage, cells are sufficiently differentiated, so they undergo multiplication, differentiation, and based upon their morphology, what is their size, what is the shape of their nucleus, their staining properties, the granules which are present in them. So based on all these structural information, we can divide them, or we can identify them morphologically by under microscope, and these cells are erythroblasts, myelocytes, monocytes, and megakaryocyte. So megakaryocyte again give rise to formation of platelets. So their vast amplification or cell division takes place at these stages, and so when we observe the bone marrow, we found these cells most abundantly in the bone marrow. Okay, and as the cells get differentiated and they get mature, they get the specialized characteristic, like hemoglobin starts appearing in the erythrocytes. Okay, we will discuss it in detail during erythropoiesis, at what stage the hemoglobin will start appearing, and then the concentration of hemoglobin goes on increasing, like other cells will have their different types of granules in their cytoplasm. So as the cells mature and differentiate, the specific characteristic features will start appearing in the cells, and after final maturation, or in RBCs, the reticulocytes are also released in the circulation, but their number will be very less. So what determines whether the hematopoietic stem cell will give rise to RBC or WBC or platelet? So what determines this? So there are various theories which explain why the particular hematopoietic stem cell will give rise to either RBC or WBC or platelet, and these theories are called as theories of commitment. So what is commitment? It is the irreversible differentiation events that lead to further limitation of the potential for development into terminal cell types. So once the cell gets committed to a specific type, then it will go on differentiating, and it will have its own features, so that ultimately the final output will be a specific cell type. So that is the commitment to a specific cell. Now what determines it? So there are various theories. The first one is extrinsic regulation. That means extrinsic means which is outside. That means the outside environment of the cell will determine what will lead to formation. That means if we have certain growth factor in my vicinity, that is called as microenvironment. So the factors which are present in the microenvironment, they will determine the final output of that cell. So that is called as commitment step is determined from outside of the cell by a specific signal or combination of signals, and most probably initiated by ligands for which the cells have specific receptors, and downstream events involve specific gene expression. That means the microenvironment will have specific growth factors, and that cell has the receptor for that growth factor. So these ligands will bind with these receptors, and they will determine whether the hematopoietic stem cell will develop into RBC or WBC or platelet or lymphocyte. So the external environment to which these cells will be exposed, they will determine the final output. And there is another theory, that is stochastic mechanism. So it says that the gene expression that establish the alternative differentiation events are stochastic. That means randomly it takes place. So these are the two opposing theories. One is saying that there is some control mechanism which is present in the external environment that determines the final output, and or either randomly it will be determined. So but whatever research we are getting is, there are certain growth factors which are called as hematopoietic growth factors, and that they will determine the final output. So we have various hematopoietic growth factors, and these are mostly glycoproteins, and they are capable of supporting a particular spectrum of cell types, and their number is already large, and many more are discovered till now. And these growth factors are, the most important is erythropoietin, then KIT ligand, interleukin 1-2, granulocyte colony stimulating factor, granulocyte macrophage colony stimulating. So this is the entire list of growth factors which have their receptors. The receptors are present on these cells. So these growth factors will act through this receptor, and they cause transcription of various proteins, and then they determine the differentiation of cells. So as I already told you that these cells have the receptors, and the receptors are mostly of two types, cytokine receptor family and tyrosine kinase receptor family. So they undergo there is whole mechanism downstream how they act, and then they will lead to formation of various proteins and then differentiation of these cells. So now we know that we have the hematopoietic stem cell which gives rise to different types of cells. Now you must have heard about transplantation of the bone marrow. So what is the means what we transplant in the bone marrow? So we need the hematopoietic stem cells which will be recovered from the bone marrow, okay, and then it will be given to the patient. Now in which conditions we need to transplant this bone marrow? So the conditions are aplastic anemia. Aplastic anemia means the bone marrow is means that bone marrow is not working. It may be because of irradiation or chemotherapy which is given to the to the patients having malignancy. So in this condition the bone marrow is aplastic, and we can the treatment is transplantation of bone marrow, that is hematopoietic stem cell. It is also done in thalassemia, sickle cell anemia. So these are the conditions where the RBC structure is affected and there is more hemolysis. So the RBCs which are present, they are damaged and means no, so they will be removed by the spleen and liver. So we need to give the hematopoietic stem cells which are structurally sound or normal, so that they lead to formation of normal cells, okay, so that there is no hemolysis further. Then bone marrow aplasia, severe immunodeficiencies, paroxysmal nocturnal hemoglobinuria, acute myeloid leukemia. So these are the cancers, lymphoblastic leukemia. So in all these conditions the treatment of choice is transplantation of bone marrow. So to conclude, we have seen today what are the hematopoietic organs, what are hematopoietic stem cells, committed hematopoietic progenitor cells, what are the theories of commitment, then hematopoietic growth factors, how they act on these cells, and what is the importance of transplantation of hematopoietic stem cells. So do you have any doubts? Any questions?
Ma'am, you said that bone marrow transplantation is done.
Huh? So what is your question?
You said that there are various reasons, diseases in which we do the bone marrow transplantation.
Huh?
But according to theory of commitment, there is microenvironment which regulates bone marrow. So exactly where we would transplant that, how we can understand? I mean, it depends into which cell it would transform depending on the microenvironment. So each person might require different type of cell to be formed.
Yes, but these are the conditions where the bone marrow is not functional, okay. So we have seen these conditions, and in all these conditions the common factor is that their bone marrow is not functional, or the RBCs which are formed, these are defective, okay. So what is done in bone marrow transplantation, we take the hematopoietic stem cell from the healthy population after MHC and after matching. Then lots of procedure is required for matching. Means we have to find out the proper donor for that person, and then if we can get the hematopoietic stem cells, then they are given systematically. मतलब like मतलब जैसे हम ये देते ब्लड ट्रांसफ्यूजन कैसे करते हैं, वैसे we give the hematopoietic stem cells, and then they will go to the bone marrow along with growth factors are also given, okay, and then they will be transplanted in the bone marrow. Huh?
If a patient requires RBCs, suppose, and we have to transfer bone marrow, then how we will ensure that those hematopoietic stem cells will differentiate into RBC only and not into some other thing?
See, that person will need every cell, not only RB. Even though if he has anemia, it is not like that he will not need neutrophil or lymphocyte or platelet. Every person will need every cell. So if we are doing the transplantation of hematopoietic stem cell for any particular reason like thalassemia or sickle cell anemia, it is not that they will form only RBCs, okay, because they will need every cell. So you will learn in medicine also, means you will learn it in detail during in medicine classes during your third year. Okay, but for now, remember that in which conditions we need to give the bone marrow transplantation. Okay.
Please explain the reasons for the quiz.
Okay, so can we do it next time during our next class? Okay, because we have another lecture at 3 o'clock. So we will discuss the quiz questions during the next class. Okay.
Please show the
What happened?
show the answer slide.
See, I have the answers, but we will discuss it during next class. Okay. Okay, ठीक है. If you have any, हाँ, I will send the PowerPoint, and if can you repeat?
Why can't committed progenitor cells form lymphocytes?
For lymphocyte we have another cell.
Yeah, why is that?
So why is that? There is no question. There is no answer for why is that. This is there, and it is observed experimentally that how the cells develop. So for lymphocytes we have lymphoid stem cell, which is derived from the hematopoietic stem
No,
cell.
ma'am,
Huh?
in the next slide when you showed that the pluripotent cell forms. Yes, ma'am, this slide. So here, why is pluripotent hematopoietic stem cell directly forming the lymphocyte and not going through the other pathway?
So there is no answer for why. This is observed over the years. The scientists have observed like these cells divide in this manner, and this is the observation, and this is the program.
And ma'am, you mentioned something about hemangioblasts. What are they? Can you repeat?
Okay, so hemangioblasts, these are the like definitive hematopoiesis starts from this place, aorta, gonad, mesonephros, and ventral surface of the dorsal aorta. So these are the cells which are called as hemangioblasts because they give rise to both endothelial cells as well as blood cells. So they are called as hemangioblast.
Thank you.
Okay.
Ma'am, is myeloid stem cell and committed progenitor cell same?
Sorry, can you
Myeloid
repeat?
stem cell and committed progenitor cell are same?
Here, yes, this myeloid stem cell, these are the committed cells to form the myeloid series. Okay, so if you have any question, then you can mail, you can send the mail to me also, or we can discuss it in the next class. Okay, so go read this chapter and then we will discuss it. Okay, bye. Thank you, sir. Thank you. Sir, I will send you that. मुझे रहने दीजिए.
Thank you.
Good afternoon, everyone. Let me see. Okay. Right. Good afternoon. So we'll carry on from where we left last time. There were certain queries, I think, in the last lecture regarding the mechanisms of homeostasis. So I will just quickly redo them. So okay. Good afternoon. Thanks for the messaging. The messaging can stop for a while. What is this showing? All right. Okay. So we start with the today's lecture, but before I start, let me touch upon the homeostasis. So we discussed that the homeostasis is primarily refers to various mechanisms that operate in the human body, of course also in animals, that maintain various parameters of the internal environment within specified range. And we can take multiple examples. It could be regulation of blood pressure, osmolality, pH, temperature, oxygen saturation, carbon dioxide levels, hormone levels, even what Dr. Gitanjali was talking, even hemoglobin, its levels are maintained within normal range by principles of homeostasis, mechanisms of homeostasis. So there are three mechanisms of homeostasis. Can somebody just type it in? Three mechanisms that we spoke last time. Lovely. So Manit, negative, positive, and feed forward. Lovely. Of these, principally the negative feedback mechanism is what one would call, the way it was described by Walter Cannon, as the principal mechanism of maintenance of something within normal range. The positive feedback mechanisms, per se, when you look at intuitively, they don't seem to be fitting in the understanding of maintenance of something as a regulated variable. However, when these positive feedback mechanisms are part of a larger negative feedback mechanisms, then they help. And lovely, Karthike, as just mentioned, part of larger negative feedback. And these positive feedback mechanisms help in amplifying the process and also increasing the scale of the response. So in that sense, it is helpful. Feed forward mechanisms wouldn't fit into the typical definition of saying that something changes and therefore as a response. So it is in a way anticipation of a change. Yes. So Banut mentions anticipation. So we can take these examples. So I'll take one examples of both of them, the negative feedback and positive feed forward mechanism, just to reiterate what I mean by them. So you take an example of let us say blood pressure. So we expect a healthy person in an upright position to have blood pressure of about 120 by 80, and let us say something happens and the blood pressure drops. This drop in blood pressure will be sensed by certain sensors. The information will be sent to the integrators, and the integrators will then give an effective response to bring the blood pressure back to the normal values. So therefore, there was a change in the regulated variable and system responded. So for a negative feedback mechanism, error must occur in regulated variable and the effect should reverse the change in the regulated variable. This condition must be satisfied before I can call something as a negative feedback mechanism. On the other hand, a positive feedback mechanisms works differently. So, a feed forward mechanism. So, in a feed forward mechanism, there is no as such change in the regulated variable. Now, let us say, let me go back to the blood pressure example. So, when you want to run, during running, you would like to have a higher blood pressure because that will help in blood supply to the muscles. And even before you begin to run, the neural signals from the brain in anticipation of running leads to increase in blood pressure. So therefore, in this condition, the blood pressure is shifting from 120/80 to 140 or 160 by 100 or maybe 160 by 80. This is useful for running, but not useful when you are at rest. So this is a feed forward mechanism which changed the blood pressure, but you wouldn't call it as a feed forward mechanism of homeostasis because it is not maintaining the blood pressure in the normal range. It is useful while running, only in that condition, but I wouldn't call them this feed forward increase in blood pressure because of anticipatory requirement as part of a homeostasis, because by definition homeostasis would be keeping these variables within the narrow range under resting conditions. This is the way Walter Cannon thought of it. Yeah. So the receptors in the hypothalamus, Jessamine, are the neurons themselves. So you have areas in the medial preoptic part of the hypothalamus which sense these. So you got cold neurons and hot neurons over there which can sense these temperatures. So their firing rate changes. So the neurons themselves are the receptors. The same thing applies for the osmoreceptor, central osmoreceptors. So when there is change in the osmolality, the neurons in a region of the brain called as the OVLT regions, they sense these changes and they are neurons themselves. So you do not have a separate sensor as such. The neurons themselves are the sensors in the brain itself. So, but then it qualifies as a part of homeostasis. So in a feed forward mechanism, the example that typically I gave you was is the core body temperature. So even when you step out in the cold, eventually the cold would decrease your body temperature, but even before the core body temperature has decreased, and therefore when negative feedback mechanisms would start, the receptors in the skin would sense it. Ajay, you correctly mentioned that. And these receptors, the skin temperature then interferes with the negative feedback mechanism which is existing. So temperature regulation is combination. It has a negative feedback component and it has a positive feedback, the feed forward component, and both of them work together. Okay. So in that sense, let us say if you were resting and a patient was infused with cold saline. Okay, the doctor did not take adequate attempts to warm the saline to the body temperature before infusing. Okay, so this cold saline will decrease the core body temperature and shivering will begin. So this would be a typical negative feedback mechanism for maintenance of core body temperature. But a cold exposure on the skin would initiate this mechanism even before the body temperature has dropped. Therefore, it would be a feed forward mechanism. Ultimately, everything would need sensors. So it is not that the feed forward mechanism work without sensors. They too need sensors. Okay. So it is the construct in which you work. Am I okay with this component? Because then I'll move forward. Negative feed forward. The negative feedback and the feed forward mechanisms. Is all right? Lovely. And we spoke of other examples. We can. So when you look around, think of your responses to what happens all around, and you'll be able to classify your responses as negative feedback or feed forward or something like positive feedback, if at all it comes in the whole construct of homeostasis. So for homeostasis to occur, we need these things: an afferent mechanism, integrating centers, and effector systems, right? And one of the major systems of the body which participates in homeostasis is autonomic nervous system. And when this autonomic nervous system was initially described in late 18th century and early 19th century, late 19th century and early 20th century, it was thought only to be as effector organ. But today we understand that the autonomic system also has large afferent components. It also acts as an integrator and an effector. And along with the hypothalamus, it puts everything together, because when we're taking the temperature example, in humans or also in animals, when you're exposed to the cold, not only do you do shivering, but you also wear warm clothes. So wearing warm clothes is a behavioral response. Okay. Shivering would be in some way physiological response by increasing tone of the muscles. So behavior is a large part of these responses to thirst mechanism. When we spoke of, is a behavioral response, and release of angiotensin II, release of ADH affects the organ system directly. What one would call as the physiological response, though behavior is also physiological response. So we discussed this. Walter Cannon coined this term, and in his initial descriptions, he took umbrage. He referred to the autonomic nervous system as an important role in maintenance of homeostasis. So what we discussed in the last lecture was the principles. What is negative feedback? What is feed forward? What is positive feedback? Now the question is, who does this? Who are the players? So the major players are the autonomic nervous system, and I will be discussing today in this lecture the small various components of it. So Gaskell and Langley, these two group of people and their friends working together, they coined this term as autonomic nervous system. But they also mentioned that it is not as autonomic as autonomous as we think, because it can still be affected by cortical processes. So it is not totally autonomous. Thoughts can affect it. External situations can affect it, and we all know that the smell of food itself can begin to cause release of gastric juices in your stomach, or salivation will begin to occur. So this is not that autonomous, that not under so-called voluntary control. It can be initiated. You can get angry at times and your blood pressure will shoot up, not necessarily because you're totally autonomous. And in their original description, they included three components to it: sympathetic, parasympathetic, and enteric. Enteric, of course, refers to GIT. However, if you read today's textbooks, they will generally not talk about enteric as part of autonomic nervous system, and generally autonomic nervous system will be spoken in terms of sympathetic and parasympathetic component, not the enteric component. But in the original description, it was part of the enteric nervous system. However, enteric nervous system as such is highly influenced by the autonomic nervous system through sympathetic and parasympathetic supplies. I will restrict to today's discussion to sympathetic and parasympathetic as a mechanism. Whether it's negative feedback, whether it is feed forward, they will operate through these neurons. So the bulk work of afferent efferent integration occurs through ANS. It operates at multiple feedback loops with several levels. I will show you these diagrams. And involuntary, we discussed like baroreflex shivering, which is involuntary. However, there are even, one would say voluntary, but more than pure reflexes, the response do occur. And what are the major systems that are affected by the autonomic nervous system? Cardiovascular, respiratory, GIT, and reproductive. Effectively, what keeps you alive? Okay. So what are called as the vegetative functions? These are controlled by autonomic nervous system. Your muscles are not controlled by the autonomic nervous system. You need separate set of neurons, motor system, to be able to go and drink water, to be able to run away from danger, to wear warm clothes. You will need the activation of motor system. Similarly, hormonal releases would not occur via ANS. You will read hypothalamus and endocrine system for that effect. So ANS is part of the homeostatic machinery. Okay. And what variables can be regulated by ANS? Temperature, bowel bladder controls, osmolarity, pupillary diameters, metabolic influence, and you can just keep on adding the list. This is not a complete list, just a few examples where we can think of autonomic nervous system affecting these variables, regulated variables. Now this is one diagram to show you the complete picture, and this is, in my understanding, the whole integration of homeostasis. As an animal, as a human body, you need to know what is happening inside your body, what we call the internal state of the body. And you need external information, where you are, the external environment. So you have somatic afferents, touch pain, which will give you some information through spinal cord. You have eyes to see what is happening around, whether it's a food, or you have nose to sense chemicals, danger, food, mating behaviors. So spinal cord will carry those somatic afferents, the cranial nerves will carry the information from nose, even from ear, eyes, and give it to the central systems. And we discussed last time, the nucleus tractus solitarius is a large afferent component system of the sympathetic and of the vagal and the glossopharyngeal inputs. Okay, so all efferents coming from the internal state. So what are we looking at in internal state? So visceral information from the GIT, meaning how much food you have taken, what kind of food it is, whether it's fat, whether it is salt. All the information are coming from the GIT. Lungs will carry information, how much stretch has occurred, whether there are irritants in the lungs. Reproductive organs will carry information as to whether it's the ovulation is occurring or other components will be taken up. So these vagal afferents, this will go for two components, visceral afferents and vagal afferents. I will be talking again a little later. The CVO, which I have mentioned, is related to the osmolality information. Then you have hormones released from the peripheral organs. They will also go up to the system. So some will go via nerves, which is the visceral afferents and the vagal afferents. Even some components of sensory system also takes part in cardiovascular, but it's majorly vagal afferents. CVO refers to, just a minute, yeah, let me just, yeah. These are the organs, these are part of the brain which are outside the blood-brain barrier. These are the parts of the brain which are outside the brain barrier and include, I'm just typing in, okay. So this includes OVLT, organum vasculosum of lamina terminalis, okay. One subfornical organ, okay. These two organs are present just in front of the hypothalamus, just in front of the third ventricle. These are neurons. Usually the brain is separated from the blood by blood-brain barrier. These two organs, these are small parts which are outside the blood-brain barrier. That means they can directly sense the hormones in the blood, or they can directly sense the osmolality in the blood. An additional organ which is very next to the nucleus tractus solitarius as well. So am I okay with this, CVO? Right. So effectively, CVO sends the directly the proteins in the brain, proteins in the blood. So in fact, let us say you are having food, and before even the food has been absorbed, from the duodenum, you will be releasing cholecystokinin, secretin, insulin, various other proteins, which do metabolic function, but they will also go to the hypothalamus. See, everybody receives blood. All neurons, Aditya, all neurons receive blood, but between the capillaries and the neurons, what we call is blood-brain barrier. This is present between the neurons and the blood in the capillaries. Therefore, large proteins cannot pass through it. However, the CVO, which meaning OVLT, subfornical organs, they do not have blood-brain barrier. That means they can sense, they have access to proteins in the blood. Yes, so astrocytes, yeah, they help in forming the blood-brain barrier. Yeah, that's correct. Sushmita, astrocytes help in formation of blood-brain barrier along with other glial cells. Okay, but the CVOs, they do not have the blood-brain barrier, and therefore they can sense this proteins in the blood. Okay, so let's redo it. How does the brain get to know what is happening in my body? Whether it's GIT, respiration, respiratory system, cardiovascular system. What are the possible ways? One way, the neural pathways. The neurons will carry from stomach, from duodenum, from atria, from lung receptors. They will carry information via afferent neurons to the brain. That is one mechanism. The second is the organs release hormones. The organs release proteins, which will go reach some parts of hypothalamus which are outside the blood-brain barrier, like preoptic nuclei, like OVLTs, and then they will be able to send that information. CVO specifically also sends osmolality. So not all neurons in the brain will be able to sense osmolality. So the internal situation of the body will go through visceral afferents, mainly vagal. Cardiovascular afferents also go to sympathetic. So it's not that sympathetic do not have afferent. They do also have afferent, but majorly vagal afferents. And these will reach to, if you see, limbic system and hypothalamus are central organizers, along with the lower brainstem autonomic pathways. I'll touch upon this. And the external information is coming through cranial nerves and the somatic afferents. Okay, so external information along with the internal information, then you will be able to give an adequate response. And what response are we looking here? Response will occur in three ways: a behavioral change, a change in endocrine output, and neural effects. So we are looking at a motor effect, we are looking at hormonal effect, and we are also looking at visceral effect, sympathetic and parasympathetic mediated. Okay, we are okay with this whole integrated mechanism. No matter what homeostasis you are looking for, it may have one or more of these components or all of these components. So when we took the example of osmolality changes, you were dehydrated, you had increase in the electrolyte concentration. This was sensed by CVO. What was our response to it? A behavioral response. We felt thirsty because of limbic system. We felt thirsty. Feeling is limbic system function. So we felt thirsty, the motor neurons were activated, the animal goes around, finds water, drinks it. Simultaneously, through hypothalamus, there was release of antidiuretic hormones. Kritik, yes, the spinal cord would be involved. That's correct. Therefore, when I write NTS, spinal cord, NTS to hypothalamus, in between them you will be talking about the medullary regions. Let me go to the next slide to be able to elaborate on the same. So let me go here. So the organization of the ANS. So you have these major organs in medulla oblongata. So the primary effector regions for cardiovascular, respiration, GIT are located in medulla. It's hierarchical. So you have the cardiovasculatory centers, the inhibitory centers, swallowing centers, everything is available in the medulla. Respiration, inspiration, expiration, everything is in medulla. Over medulla there are pons. So you have higher levels at pons. Then you have higher level at mesencephalon. Then you have higher level at hypothalamus. And finally you have higher level at the cortical neurons, what is called a central autonomic network. So in effect you are looking at cortex down to medulla in a hierarchical manner. Okay, so therefore when we say point number 2 on the left, it operates at multiple feedback loops with several hierarchical levels control utilizing CNS and PNS. So you are correct when you say, is medulla involved? Yes, it is involved. Is pons involved? Yes, it is involved. Is midbrain involved? Yes, it is involved. Is hypothalamus involved? Yes, it is involved. Is cortex involved? Yes, it is involved. Like cognitive function. Now let me give you a situation. A person is asked to sing in a public fora. Suddenly your blood pressure is running. You begin to tremble. This is a cortically driven response because there is no actual fear. Nobody is going to eat you up. But the whole machinery of fight flight gets activated because cognitively we start thinking, oh, I'm in a, so many people are watching me, and you create that situation as a stress situation. Actually, there is no stress as far as life is concerned, but the systems, you've been trained cognitively to think it as a stress, and therefore you begin to respond like a stress. So this is a frontal lobe cognition driving the limbic system, driving hypothalamus down to the medullary regions. So yes, medulla would be a part of the whole system. All right. Now there is a sort of behavioral partitioning, not completely exclusive partitioning, but major behavioral partitioning. The sympathetic system usually tends to get activated during stress and adversity, fight, flight, and certain mating behaviors, and parasympathetic tends to be activated for digestion, growth, and repair conditions. But that is not necessarily exclusive. Both systems can be simultaneously activated or simultaneously inhibited. And I'll be taking one lecture in the very last component as to this integration of mind body, where I will touch upon this again, as to in what conditions of mind body interactions wherein you have activation of both or deactivation of both. Okay. So back to the afferent pathway. So we spoke of the afferent components of ANS would be the neural. So in this diagram, they were internal state, went through hormones, through direct sensing, and you had visceral afferents and vagal afferents. So this is, so the neurons will be called as the afferent systems. So majorly the visceral nerves, which comes from your GIT, from cardiovascular system as well, they will go to the lamina 1, lamina 5 of spinal cord. You need not remember this right now, as and when you will discuss the CNS, this will come back again. Just for your registration, just keep it over. Even if you forget now, it's okay. And this 9th and 10th, 9th of course the glossopharyngeal. So upper part is carried by glossopharyngeal, mainly from the pharyngeal regions, and from the carotid regions the afferent information from the cardiovascular and GIT is taken by glossopharyngeal. The rest of the body down will be carried by the vagal afferent. And if you see the afferent to efferent ratios in vagus, 10:1. That means for every one effector fiber, for vagus there are 10 afferent fibers going up, indicating the importance of the afferent information. Without information, you can't get a good control, good regulation. So afferent fibers are large part. And what do these carry? I'm repeating. Blood pressure information, blood volume information, how much is oxygen in your blood, how much carbon dioxide is in your blood, how much distension of GIT has occurred, what hormones have been released from the GIT, bladder information, food content, the calorie of the food, the protein, fat, other chemicals in the food can all be sensed. Bladder distension, hypothalamus directly can sense. Somebody had earlier asked the question, what about hypothalamus? Yes, the neurons in the hypothalamus can directly sense these glucose and osmolality and temperature. Then you have eye, which will give you the external threat information, and nose obviously smell, and of course ear can also be includes different kind of noises can make you respond in that sense afferently. So this integrating slide that we discussed, and you will see that on the right side there is a sort of a segregation in the brainstem area. Certain group of neurons responding to heart, cardiovascular system, some controlling bladder function, some controlling digestive function, breathing, sexual function, muscle tone. Obviously it's modular but all integrated. And who will integrate them? Higher centers. Who will integrate with them with other functions? Higher centers. So it's a total integrated function from top down, hierarchical. The efferent component, let's look at that. So traditionally we classify them into sympathetic outflow and parasympathetic outflow. The sympathetic outflow, I'm going to show a diagram which you can find in almost all textbooks. So I've copied from one of the textbooks. As you can see over here, from the brainstem onwards, different organ systems are affected or supplied by the efferent pathways. So the color coding has been done. So the parasympathetic, what we call as the cranial outflow, because it majorly goes by the cranial nerves 3, 7, 9 and 10, shown in that what color they call it, violet, yeah. so they're violet color, and you have the sacral outflow coming from the lower part, that is the S234 segment of the spinal cord, going to bladder and genitals. The sympathetic outflow comes from T1 to L2. So the connection will come from the brainstem, it will go down the spinal cord, and from spinal cord it will go out from T1 to L2. This will be the sympathetic, through sympathetic chain. Again, these details you will see repeatedly in anatomy lectures, and also as you read or as you taught each individual organ systems. So this is a good composite diagram. You can check any other diagram from any other textbook. All are almost similar, but major idea is this. I'm sure all of you are perhaps aware of it. I do not know whether they teach this in now in school days or not, but so are we okay with this effector system, the diagram? Lovely, Sagar. So right, can I move on from this? Yeah, Alicia, thank you. So now this is a little more expanded version of the green part which is coming out of the spinal cord, this part, showing you how each segment of spinal cord leads to these neurons. And I'm going back to the slide, previous slide. So you see there are these efferent pathways which come from the either from the brainstem or from the spinal cord, are two neuron systems. There is what we call as a preganglionic neuron and a postganglionic neuron, and these two synapses somewhere in between. When we talk of sympathetic system, the synapse occurs in the ganglia, what we call as a sympathetic ganglia, and they are away from the organ in which they to which they supply. While if you see the parasympathetic, you can see they are very close to the organ, almost in the organ itself. So the preganglionic fiber of the parasympathetic actually reaches the organ, where it synapses with the postganglionic neuron, and the pathways are affected. While if you see the sympathetic component, you see the dark green and the light green. The dark green is the presympathetic, preganglionic sympathetic fibers, and the light green is the postganglionic sympathetic fibers. And the sympathetic ganglia are aligned along the spinal cord in a paravertebral region anatomically, while the parasympathetic ganglia are usually very close to the organ in which the postganglion innervate. Therefore, the postganglion fibers are very small. So, for example, the preganglionic fibers for the heart would reach the heart, and the postganglionic fiber will be in the heart itself. So will be the ganglia. While the pre-sympathetic fibers, pre-ganglionic sympathetic fibers for the heart will synapse in sympathetic chain, and the postganglionic fibers will then reach the heart. Okay. So this is a diagram just to show. Karthik, I don't know whether it served any purpose or not. No way of, I don't know whether having ganglia of parasympathetic next or very close to the organ has a benefit. There is no way of answering. I mean, one can speculate, but very difficult. I mean, I have no idea as to what advantage would it give, except for maybe if I were to speculate, that the presympathetic ganglia allows for some level of integration, because it can get afferents, and some level of integration can occur at the ganglion level, because fibers can go up and down, maybe in that sense. But otherwise, I don't see any benefit up front. Don't know. Why meant how? So somebody correctly pointed out, let me just see who pointed it out, that not to ask why. Why can be asked. Why can be asked, but as a physiologist I won't be able to answer. You can ask why. Why is philosophical? Jessamine? Okay. But difficult to answer these questions. Why? Okay. So back to the sympathetic outflow. So this is the spinal cord, and you can see what we call as the IML. This is the anterior part, the posterior part, and intermediolateral horn of the spinal cord, from where the preganglionic fibers of the sympathetic system arise. They go through connections to the sympathetic ganglia, and then they pass through various visceral nerves to various organs. And on the right side is a diagram showing the same thing. Okay, the chain of ganglia next to the vertebral column. So you can see the spinal cord, and there's a small chain of these fibers next to the vertebra, sympathetic chain. Okay, so people generally tend to compare sympathetic and parasympathetic. So I've taken this from one of the textbooks. Almost all textbooks would be giving similar information. I found this to be a little more comprehensive. So let me read it for you one by one. Sympathetic system originates in thoracic and lumbar region of spinal cord, T1 to L2, while the parasympathetic originates in cranial nerves and sacral regions, what we call as the cranial outflow and the sacral outflow. So the ganglia are located in paravertebral sympathetic ganglia or the collateral ganglia, while for parasympathetic system, the terminal ganglia are located or near or embedded within the target tissues. Obviously, short fibers and long fibers. Please note, for both the preganglionic fiber in the ganglia and the synapse, they both release acetylcholine as a neurotransmitters. Whether it is sympathetic or whether it is parasympathetic, both preganglionic fiber release acetylcholine as the neurotransmitters, while the postganglionic fibers release, they are what we call the adrenergic, and they will release norepinephrine as the neurotransmitters, while the parasympathetic, both the preganglionic as well as the postganglionic release acetylcholine. And to some answer which was asked, possible reason why it can happen: divergence coordinates activity of neurons at multiple levels of the spinal cord. Parasympathetic has limited divergence, so maybe in some way it could help, but then there's no way of knowing that why is very, very difficult to answer. How we know why it's difficult? And sympathetic system, kindly note, activity often involves mass discharge of entire system. So when sympathetic system is activated, it generally tends to get activated in a larger space. So the whole of cardiovascular system, along with respiratory system, along with GIT will be activated, while parasympathetic will have a very, very narrow range of action. So very discrete organs. And the primary neurotransmitter postganglion, NE norepinephrine, postganglion is acetylcholine. Sympathetic predominates during emergency fight-flight reactions and predominate during the quiet resting conditions. So these are something sometimes your teachers and your examiners do ask in exams, to contrast and compare sympathetic and parasympathetic. So this is the slide that can help you in write those exams. But conceptually, I hope you have understood this. Now, there are 3 slides which I'm going to show you. This I have taken from Ganong. Again, similar information is available in all textbooks. These are very difficult to remember as such or at one go. So it shows that if I were to stimulate a specific sympathetic fiber, what action will it do on the target organ? So in adipose tissue, lipolysis occurs. Parasympathetic, no supply. In adrenals, it causes increase in secretion. Parasympathetic, no supply. In the eye, you can see a contraction, but in ciliary muscle causes relaxation. Please note this. In radial muscles, contraction, ciliary muscles, relaxation. On the parasympathetic side, it's the reverse, relaxation and contraction. Therefore, the effects are organ specific. It will not be appropriate to say sympathetic leads to all increases, not necessarily. Parasympathetic leads to all decreases, not necessarily. It is preferably you should ask which organ are you talking about. And even within the same organ, you will find that a sympathetic can lead to an increase or decrease depending upon the receptors present on those organs. Okay, so let me shift to next slide. You see, look at the islets, pancreas. Increase in secretion, decrease in secretion, depending upon the receptors present. You look at bronchial glands, increase in secretion, or both caused by sympathetic effects. Okay. So, if somebody were to ask me, what is the effect of sympathetic stimulation on pancreas or islets of pancreas? Say, tell me which receptor it is acting upon. So, it is the receptors which will decide the effect of norepinephrine. Okay. So, for the slides, so this is a compilation I've taken from receptors type A and type B. There's a classification of it. So, as you will go through the various organ systems, so when somebody is teaching you cardiovascular system, they will give you details of alpha receptors, beta receptors, even for acetylcholine, muscarinic receptors, nicotinic receptors, they will give you details. So, if a question is asked to you, it's an information based question. What is the effect of sympathetic stimulation on stomach? Then I must need to know the receptors present in the stomach. So in the stomach, it leads to decrease in motility and tone, but causes sphincteric contraction. Okay. So body relaxation, sphincter constriction. The same sympathetic system, because of difference in the receptor location in these organs. Okay. So it would be inappropriate to say sympathetic leads to contraction or increases while the parasympathetic leads to all decreases. No. You look at the urinary bladder. The sympathetic stimulation leads to relaxation of detrusor muscle. Yes, receptors are norepinephrine. That's correct. Because once the neurons will release new norepinephrine. So whatever is present in the receptor, it will act upon. So if it's a beta receptors, generally will decrease. Alpha receptors, it will increase. And as you will read pharmacology, we give different kinds of drugs, though they are sympathetic stimulators, but they stimulate either alpha or beta, and accordingly their effects are produced. Alright? So this particular the compilation is something which is I have taken from, I think, Genome. We could take from any other book, and the things will remain the same. You need to have that information. There is no I would say logic in it. It's information, memorize. You got to memorize these things. And if you don't use this drug repeatedly, you will forget about it. I also forget. If you suddenly ask me what is the effect of A, B, C on what receptors, I say let me check it up. Don't rely on memory when you have today devices to access information. Memory is not something that you should pride upon. Ability to understand things that you should pride upon. So what is the difference in sympathetic innervation to skin? Yes. So the neurotransmitter which is released in the skin from these sympathetic fibers also causes acetylcholine release. So that's an exception. Somebody nicely pointed out. So everywhere it is. Actually, if you go in further finer details, many of the autonomic nervous system sweat glands in the skin. Around there, sympathetic fibers will lead to release of acetylcholine. So cholinergic sympathetic fibers. You will also have, if you again read minor details, autonomic fibers which release non-adrenergic, non-cholinergic neurotransmitters. So you could have a nitric oxide or vasoactive polypeptides. So these are, apart from this, there are many other neurotransmitters which are released. So it is what we call as NANC. I'm typing it out. NANC, non-adrenergic, non-cholinergic fibers of ANS. Okay, this variety also exists. However, majorly sympathetic postganglionic fibers will release norepinephrine. Majorly postganglionic parasympathetic fibers will release acetylcholine. Okay, exceptions do exist. Therefore, calling the whole autonomic nervous system as one framework may not be completely true. Largely yes, but not in nuances. Okay. Nicotinic receptor, nicotine cholinergic. So there are two groups of cholinergic receptors. Nicotinic and muscarinic. Okay. So in the ganglia of both sympathetic and parasympathetic, the preganglionic fiber release a metabotropic is different kind. Is a different kind. So now we are now getting into the receptor classifications. So receptors. Okay, Jaseman, let me repeat. Let me elaborate rather. So in the preganglionic fiber, both of sympathetic and parasympathetic, what will be released in the ganglia? In both the ganglias, acetylcholine is released. Right, correct. But the receptors on the ganglia, which one are these? These are nicotinic acetylcholine receptors. They are different kind of receptors. Okay? In ganglia. The post-ganglionic parasympathetic fibers will also release acetylcholine, correct? There, the receptor is muscarinic. Different receptor, different kind. Even in nicotine and muscarinic receptors, you will have classifications. So there will be M1 to M5. So M1 to M5 receptors. Subclassification. Smoking increases adrenaline level. Not necessarily actually. I mean, smoking per se won't release which one? Gautam, I don't get the background of your statement. Smoking increases adrenaline level. See, we do not have any pathway which per se leads to the chemicals in the smoke. Irritation leading to tension leading to adrenaline release can occur. It is related to nicotine. That's why the name came. But it is not the nicotine we kind of smoke. Okay? What we smoke does not go to the ganglia. What you smoke stays in the lung, get absorbed, stays in the blood, has its own effect on nicotinic receptors everywhere in the body. For that nicotine that you have smoked, for it to reach the sympathetic ganglia and parasympathetic ganglia, very, very less. Kartik, you are correct that the postganglionic fibers have nicotinic receptors. That means these receptors respond to acetylcholine released by the preganglionic fibers, and they are called nicotinic because they were found to be also stimulated by nicotine. So if I put nicotine in a parasympathetic ganglia, if I put nicotine in sympathetic ganglia, both will lead to activation of postganglionic fibers. Okay. See, all these names have taken. We go back 100 years. Some scientist was having nicotine in their hand, and they injected the drug and found some effects, identified those receptors, and they call it, oh, this is a nicotinic receptor because it responds to nicotine. Therefore, they gave it a name. That does not mean it does not. When they actually found out, oh, nicotine is not released in the ganglia. What is released in ganglia is acetylcholine, and the receptor present on the postganglionic membrane also responds to nicotine. Okay, but that doesn't mean body makes nicotine. We don't make nicotine. That's correct. Jaseman, both can cause. And since whether it is sympathetic or whether it is parasympathetic, doesn't matter. If you put nicotine in the ganglia, there will be a increased discharge both in parasympathetic as well as sympathetic postganglionic neurons. Acetylcholine, Anaga acetylcholine. Let me just Google up for you. That will also be helpful, so that you can see on the screen. So let me write down receptors for acetylcholine. Okay. Let me see which is a good, which you can easily see. I'm taking Wikipedia. It's not a bad sometimes starting source. So is this available? Can you see this Wikipedia online?
Yes, sir.
So let me go to the receptor subtypes. Okay. So you have NACHR and you have the muscarinic type. It's a classification. Okay. So I won't say trust Wikipedia. This is not your primary source, but just for my ease, I have taken up this from Wikipedia just to show you that you have nicotinic acetylcholine receptor and you have muscarinic acetylcholine receptors. Okay, let me just click on the muscarinic acetylcholine receptors and show you the different types of classification. So if you see here, I'm just scrolling down. Can you see M1? Then M2, then M3, then M4, then M5. Okay, and if you see on the right side, there's a huge list of various agonists and antagonists, and you see also the effectors, what exactly these receptors do. So the same acetylcholine will have different effect depending upon which receptor was present on that particular organ. Okay, so the whole list, and you can see various toxins affecting it. Some affect M1, some affect M2, some affect M3. It's really difficult to remember all this. Okay, as long as you get the concept right, you will be able to understand. And many of them are drugs used for treatment. We used muscarinic drugs, we use carbachol, methacholine. We use all these drugs we use for treatment. Okay, pilocarpine is utilized for treatment in the eye. Okay, so all these drugs are useful. So when you will read about pharmacology a little more, the basic idea that you should keep in mind would be that do not go simply by whether it is NE or acetylcholine. You start looking at what receptor is getting stimulated. Okay, and I would like to explain this by analogy. So that's not the best way to explain it, but analogies help. A whistle as a signal can be understood differentially as to where the whistle is being blown. If it is in a school, it means something. If it is in army, it means something. If it is at a railway station, it means something. If it is in public fora, it means something. So the whistle, the same whistle will have different effect depending upon the recipient. Similarly, the effect acetylcholine has on different organ systems will depend upon the receptors present there. So M1 to M5, and they have their different varieties, their outcomes. Let me go to the, just show you the this one, nicotinic receptors. Again, I go to different types of receptors, bindings, and their functions will be given. Okay. As of now, you can hold on to this large amount of information. There is no need to remember them, as you will go through different organ systems. Oh, I'll have to find out. I don't remember, Alicia. I don't remember exactly what happens on the mast cell. Alicia, I don't remember the receptors on the mast cell, so I won't hazard a guess. Basis of classification: Receptors have been classified initially on the basis of the what stimulated them. So they found that, say, atropine affects all receptors, but another chemical affects only one but not the other. Therefore, it is more on response type. It is not a classification based on structure, based on the effects produced. Yes, acetylcholine affects all of them. It affects all of them. How are the, I'll have to go one by one. Okay, you're talking about in the slide. Okay. Mast cell over here. Decrease in degranulation. This basically means, yeah. So, I was thinking about the which receptors, alpha or beta, I don't remember that. So, what is a degranulation? So, when mast cell are stimulated, and when do mast cell get stimulated? When IgE interacts with, correct. Manit, that's correct. Mast cells secrete histamine by degranulation. So during sympathetic stimulation, that degranulation is decreased. But I don't remember whether it is through the alpha receptor or beta receptor. That I don't remember. I have to check. Let me just check it up in any case, since we are on the net. Might as well use it. Receptors on mast cell. Okay. For norepinephrine. Okay. So let us see those receptors, and this seems to be alpha 1 receptors. Alpha 1 receptor on the mast cells. Okay. So this is something. This is information impossible to remember, at least for me. Those who have good memories can remember it. I can't remember all the information. I use technology to help me remember things by taking it out. That's why we have all the technology. Okay, so back to this slides. So what was the question? I did not see that question. Nicotinic receptors, yes. Muscarinic, muscarine. There is a compound called muscarine. Let me just show you what muscarine is. Okay. Natural product from mushrooms. Okay. That's why the names have been given. See, names were given on the basis of what extract from which particular plant or something led to an effect. Okay. So almost all the drugs that you will find have origins in plant products, and then later, of course, with the chemistry we have modified them. But the origins will come from them. Okay. I mean that's the beauty of. I love the internet and the access of information. I will love to be a student in today's time to get all this information. Okay, but mind you, you will be better advised to understand the information rather than have the information. No, nicotine does not respond to epinephrine. Nicotinic receptor do not respond to epinephrine. Okay, so the idea which I am trying to tell you is that this listing which I showed you is a listing taken from Ganong. It is a very small listing. You can just keep on increasing with more details. Details will help you when you need them. Find them out. Take it out and find them. Do not try to remember all the drugs, all the receptors. It is virtually impossible. And why do it when you have technology? Okay. I mean, that's the reason I don't remember everybody's phone number. I mean, I have a mobile phone to do the job for me. As long as I know whom to call and when to call, my job is done. Similarly, as a doctors, do not take pride in remembering things. Take pride in understanding things so that you can initiate appropriate treatments. Yes, Pratik. they tend to ask. I won't tend to ask, but yes, your examiners may. So unfortunately, that's the reality of our systems. Not everything, not everything. Some part of it, yes. Not everything, not the details, but some part, unfortunately, yes. So do that as long as you require. For I don't have to give exams, so I don't remember them. You may have to remember. So you remember and forget. So do not rely upon memories, please. Never. So if see what you do daily, you will not have to remember. It automatically come to you. If what I don't do daily, there's no point remembering it. I would just take out a tab and remember it. So if you ask me what is the dosage of a drug, if I prescribe that drug every day, I'll remember it. If I don't, Sushmita, yes. Gurkirat, as of now, no. We do not allow technology. I would if I could. I will do that. Sagar, I will do that. I will send all these PowerPoints on the Saral platform so that you can see. I will do that. Okay, so let me just finish this lecture for now. So, why do we, as a physiologist, we want to know about the autonomic mechanism? What if these systems fail? Okay, so this is a slide just showing you various disorders that as a doctor you will have patients coming to you, and this is because the autonomic nervous system has failed. So let me take the first example of orthostatic hypotension or dizziness. A patient is lying down and says that whenever they stand up, they begin to have dizziness. This is because the blood pressure drops as you stand up, but the autonomic nervous system is not able to respond to it, and patient has this falling. And there are various disorders, names are given: Streidrager's, Rayleigh, drug-induced autonomic dysfunctions of diabetes, alcoholism, Parkinson's disease. Then you have bladder incontinence, nocturnal diarrhea, fetal incontinence, impotence, gustatory swellings, gustatory sweating. So in all these, there is a problem of autonomic nervous system which manifests as trouble to the patient. Okay, this is just a list to show you that whatever I was trying to teach you has some clinical implications. People ask, what if you did not know about autonomic nervous system and you saw these patients? You may be able to treat this patient by hit and trial or by rote memory, but perhaps you may not be able to understand what is happening. So once you know the mechanisms behind, you will be a great doctor. Without it, you will be maybe a good doctor but not a great doctor. Another component which I'm sure all of you are aware is placebo effect. What is placebo effect? Anybody? Placebo effect? What is placebo effect? Manit, it is not actually dhoka to the body. I mean, it is part of your regulatory systems. So, what happens is, especially that you ask, who told dhoka was, who told Manit? You can ask Manit. Manit, would you like to explain what is dhoka to the body? Betrayal. So, Karthik and Manit, you wouldn't call it a dhoka. It is a top-down, let me say top-down influence of cortex on lower ANS. Okay? When we were discussing, it actually is not fooling. So let me give you a condition. Let us say you were very thirsty. I'll come to that. Saik, I'll just come to that. Let me give you a scenario. You were dehydrated and you were looking for water. Karthik, I'm explaining that. So you were dehydrated in a jungle environment. You saw a river and you thought you should drink that water. As you were approaching that place, you saw a tiger, and a tiger is stalking you. So now what happens? Would you still go or would you stop? No. Absolutely. So at one end you have a negative feedback mechanism telling you to go, overrun by cortical survival instincts. Okay. Maybe survival. Difficult to say feed forward because it is leading to survival but not anything of homeostasis. It's just holding it back. Now let's take another example. You have pain in your limb and therefore you're just sitting around, not walking around, because if you put a foot down, it pains you. Now again situation, a tiger comes. What happens now? You forget all the pain, start running. So did you actually stop feeling pain, or cortical systems in your body initiated what is called as a central analgesic system is activated? So it temporarily blocks the information coming from the painful areas to the cortex, so that you can concentrate on survival instincts. As a result, you will find these stories of people, despite so much of pain, soldiers fighting and carrying on. Yeah, you could use these terms, Tanay, as mind diverted, or you can use any phrase. But this is part of your body. So it exists in your body. It is not something which has come from outside. These are survival mechanisms. Survival mechanisms. Temporary though. Eventually you'll have to drink water. So the limbic cortex with prefrontal cortex will make an assessment and override the lower level brainstem level responses. Okay. And therefore what we call as mind body interaction. Okay. And this is something which is real. It is not something which is fake. So the trick is in activating it. Okay. Same thing. Placebo is what causes placebo effect. Anaga? Placebo is what causes placebo effect. Let me give you a real life example as to how human beings behave. No. Manit, no. Placebo is real thing. Placebo is real thing. It does have an effect. Let me give you a behavioral example of all of us. So, how many of you use the same pen because you feel that if you use the same pen, you'll be able to give exam in the same way? So, you have a very, very nice exam. All of us. Or you have your lucky charm and okay, Jessamine, say I don't. Okay. Yeah, so all of us vary, and you will find that, and there's a background to this. It is not something Sayak has a different approach, jisme ink ho chal jayega. So in real life, you will find that we tend to repeat the things which works, and it makes logical sense. Why? Because in a wild environment, familiarity is useful. Familiarity is useful for survival. Same environment which you have already experienced, you didn't die there. Same food that you took didn't kill you, so you are more likely to eat the same food in an exploratory world. Is everybody on board with me on this? Okay. So familiarity is something which is inbuilt in us, and it makes survival sense. So when people say eat with family, so when you eat food with your friends, you eat more. It is because the cortical influences are in a way geared that now there is no danger, therefore you can eat food in a comfortable environment and you can eat more food. However, if you are tensed, if the environment is not conducive, you do not know some danger may come, so you are very worried in eating, so your stomach size decreases. Okay. Good, you have Akram, you got good friends. Okay, so all combination. So in animals it is primarily survival. Let me since we are on it, if you agree, let me give you another example of mind body. When should, I'm asking a question, when should your blood pressure increase in natural circumstances? Natural. Okay, natural circumstances. Animal world. In animal world, danger. Okay, danger, fight or flight. Okay. However, human beings have extrapolated that thing into everything else. So not answering phone calls for 5 times, cross-linked to the same responses. You should not. I mean, there's actually no danger, but your brain mixed into a dangerous condition, and therefore what should occur for few minutes keeps occurring for the whole day. And if you see these videos of the National Geographic or BBC or Discovery, there is a herd of deer and there is a carnivore. The attack occurs for few minutes, some die, Animal Planet, one animal is killed. After 10 minutes, all animals are again grazing. You notice that? So the animals run for about 10 minutes, one of them gets killed, the rest of them start grazing immediately. That means the stress response occurred only on danger, not before, not after. Animal world. Now human beings, yeah, so what has happened is the very creativity that we pride ourselves, humans pride ourselves upon, on abstraction. So what made us human also gave us that vulnerability, that because we can think of future, we can plan, and therefore we cross-link our natural responses to artificial situations. As a result, going before the exam, our BP and heart rates are high. It should not. I mean, giving exam is not a life-threatening condition, but we have made it into a life-threatening condition because of culture. Very important. If you don't clear this exam, God knows what will happen in your life. Okay. So cortically, we initiate responses which were not supposed to be initiated. Okay. So it is this cross-linking which leads to this, and many of the placebos affect from the same pathways. If you trust something, you feel good, therefore your autonomic nervous system is cooled down. So there will be a person who is very so-called cool because they don't sense the danger, while somebody else is actually sensing a danger, while there may be no danger. So you could have a person who responds very excessively to a condition because they perceive danger. There may not be any danger. And there is a person, even though there is danger, does not perceive danger. So you vary from so-called cool to so-called tense. So it is your cultural linkages, which lead to all these kind of cross linkages, and large amount of behavioral therapy. Behavioral therapy is based upon this, is utilizing these principles which are inbuilt in you as a human being because of the cortical connection with the autonomic nervous system, and those links which are formed either are strengthened or broken by behavioral therapies. And many of the placebos affect because you believe they will affect. You believe, okay? So the trust factor is extremely important. So that you are in tension and somebody you trust comes and says, don't worry, your exam will go good. You feel better. You don't feel better because there is data on it. You just feel better because you feel better. But on the contrary, if somebody you did not trust came and said the exam will be better, you will say, oh, ab to pakka kharab hoga. It makes you more tensed. So your response changes because what level you attribute to that information. Okay. So that's where the placebo effect, working through, I'm just reading it, interactions of central autonomic network that involves everything from hypothalamus, cingulate cortex, insular cortex, thalamus, and they then influence the autonomic outflow, making you either so-called Rushit, to large extent, yes. Placebos, therefore, sometimes, what acts as a placebo also works on cultures. Different cultures, cingulate is a part of cortex, cingulate gyrus, limbic, part of limbic cortex. Okay, so it's like a particular kind of meditation affects on A group, not on B group, simply because the A group thinks it is part of their culture, B doesn't think it is part of their culture. So the effects are different. But these are majorly, no, placebo is not medicine. So let us say you have headache. Okay, let me just show you one article. Okay, I'm just writing placebo effect on headaches. Okay, see, please read this. This is WebMD. WebMD is a very trusted source. Okay, WebMD is a trusted source. Okay, I'm just dropping this. So WebMD is a very trusted source, and please see, I'll just show you one data. And when we say science means data, okay, almost 40%. Just look at the data. So patients' expectations have, so majorly this relates to pain, discomfort, attributes which are cortical. Feeling good, not so good. Whatever is cortical is largely affected by this. I mean, you can't give a placebo to get your bone fracture to be repaired. It won't do. Or you can't decrease your fever by placebo. But the feeling component can be very, very largely affected by placebo. Okay, so Karthik, correct. So if you go to a doctor whom you trust and the doctor is very famous, you think it's working. But it works only in the domain where the mind comes into picture. But if you got a liver damage, it won't change. Liver damage will remain the same. Yes. So if I had a cut on my skin, you have to suture it. You can't placebo it. Okay. If I have low oxygen in my body, I have to give oxygen. But how I feel because of low oxygen can be largely affected by how I feel about low oxygen, can be affected by placebos. I mean, I won't call it a positive or negative attitude, Mohit. It's not positive. It will vary for everybody. To large extent, yes. जैसे मन example, let me think of. Let us say you have stomach discomfort. Okay, and you're not feeling good about stomach, and I just give you a glass of warm water, and I don't tell you what I'm adding, and I add maybe just salt, a colored salt, NaCl, warm water NaCl, and you trust me, and I say have this, it will make you feel better. If you trust me, actually you may feel better even though nothing has changed in the stomach. It's just the same. Abdomen is still the same, but your feeling of discomfort changes, and you will say, wow, I do feel better. But nothing has changed as far as the disease process is concerned. वो आपके पेट में वही प्रॉब्लम है. Is that okay, Jaisimon? No. Placebo tends to be temporary. So it has waning effects. It does wane. But again, please remember, placebo affects those components of which are feeling components. It does not change the real biology. Okay, so it is not that I have got kidney stone. Kidney stone is not going to go by placebo. But the pain due to that kidney stone, I may feel more or less depending upon placebo. Okay. But it won't change the process of stone formation. That will remain pure biology. Okay. So as a physiologist, I wouldn't call placebo as fake medication. I would say placebo is part of your mind-body interaction. Mind-body interaction. Your state of mind is affected by perception. State of mind can affect ANS and therefore outcomes, therefore body functions, in that sense. But again, it is a psychological effect, yes. Feeling. Yes, Gautam. See, it's important for as a good physician to understand these things. Okay, if somebody feels better, they feel better. I mean, you have to, as a doctor, you should understand that it's just not A plus B equal to something else 2 plus 2, because you're looking at human being wherein perceptions do matter. Possible? Durgam, possible? Possible. I mean, to a large extent, what is counseling? Counseling is, in that sense, perceptual change, which makes you. But again, there's a limit to this. Okay, I mean, if I have fever, I can't meditate and get my fever down. You can't. Fever will remain, but how you feel in that fever can get affected. Fever won't change. The temperature won't decrease. Temperature will remain the same. Whether you meditate or you take placebo, temperature will remain the same. But your feeling of that temperature will change. See, I mean, it's a scam when you make money out of it. Jaisimon, which one? I missed that.
Sir, I asked whether placebos can have a physiological
Yes, it can.
Can it?
Yes. So,
Because it has effect on ALS as you told.
lovely.
So there must be some physiological effect also, right?
Yes.
Not just mere psychological ones.
Yes, it can manifest. And the example which I was giving you, that if you are giving nutritional therapy to someone, okay, the same amount of food that
Okay, sir.
if you eat same amount of food in a condition where you're comfortable and in a condition where you are stressed out, tensed, okay, because of the ANS effect, your absorption will get affected, your digestion will get affected.
Yes, sir. Yes, yes.
So therefore it will have an effect in that sense. So it
Can this effect also be positive? Like, can it also help give a hand in curing that particular any infirmity or something?
see that's where the whole research is occurring. Can it lead to cures? Now let me take an example of hypertension. Okay. Now
Fine, sir.
hypertension is caused by one of the which is stressful environment. Okay. So normally my BP should not increase, but I get angry, I get I'm afraid of my results, I have got a date, I lost money, and my BP is jumping every 5 minutes. So when it happens over a period of time chronically, okay, I will end up with a resetting of my blood pressure. Take another example. When you are stressed in an animal world, when there's a tiger after you, your blood glucose rises. Okay, so blood glucose increases during fight or flight. Is it okay?
Yes, sir. Yes. Okay.
Okay, because of cortisol. Because of cortisol and sympathetic activation.
Yes, sir.
Okay, this is a natural process which happens. This is physiology, correct?
Yes, sir. Yes, yes.
So now imagine you are just sitting and you got a message that the exam in which you had copied, you've been caught. Now what happens? Tensed. Apps. सागर को तो मैं लॉयर बनाऊंगा, हैं? ठीक है? So back to this. So you get tensed. As a result, your blood glucose rises. Correct? Now this blood glucose rose even when there was no threat. Imagine this keeps happening to you every 10 minutes, every half an hour, every 1 hour, for weeks on end. Because you as a personality start reacting to everything. So you get a chronic glucose overload. And then you may become diabetic. Okay?
Fine, sir. I can understand. Thank you, sir. Thank you so
Right?
much for this.
So yeah. Okay, long day for you, me.
Yes, sir.
Can we close for today? Is okay?
So I have some doubts. I read a text from Guyton yesterday, so I have some doubts.
Yeah, please. Yeah, please do.
So I didn't understand the term adaptive control for sleep. Is it the same as feed-forward mechanism or?
People use that term in that sense. Yes, they do. Adaptive control. Yes, they do.
Means it is exactly the same or kind of.
See, it's a phrase utilized. I won't use that phrase. So people, biology is not that way exact science. Okay. So different people use different terminologies. So they use it like an adaptive control. For example, let me give you that when I am running, I need my BP high. Okay. and that BP must be maintained at that 160 level. But my my Abhyuday in soon, it's already ended. Abhyuday is wanting to go. So so my BP is high at 160, and my barrel reflex must operate at that level as well. So even though my BP shift from 120 to 160, my barrel reflex still operative. So what would you say is adaptive control? The control points have shifted from 120 to 160. In that sense, you could talk about it.
Sir, adaptive control is a delayed negative feedback. What is the significance of this statement? Like, I don't get it. Even
I have to ask Gatan. I don't sort of understand what he's trying to mean. The author of Gatan expired a couple of years back. Adaptive control as a delayed negative feedback. No, I wouldn't agree with that. I don't understand what he's trying to say.
Okay, fine, sir, no problem.
Right. Thank you.
And sir,
Yeah.
reproduction in homeostasis, what we have to consider for that?
So, if you look at species level, then you would say maintenance of species. If it is homeostasis, then reproduction is part of that.
Okay.
In that sense, though Walter Cannon wouldn't agree with that. In Cannon's description, homeostasis was meant to only for human body, not for species.